Kristen Atchison here, and we're talking about Chapter 3, the biological context of development. So first I want to get some of the genetic foundations out of the way. This is not a genetics class, but it is important to kind of know the building blocks and which development is built upon. So let's just get some terminology out of the way. If you're not familiar with it, phenotype. Phenotype is the directly observable and behavioral characteristics that are a result of the genetic code. So it's what you see. So you may have genes for something that aren't being expressed. That's because your phenotype is only expressing one thing. So my daughter is right-handed. Her phenotype is right-handed, but I know that her genes have both genes for left and right-handed because I'm left-handed, so I know that she's got a gene for left-handed, although her phenotype is expressing being right-handed. Where a genotype is that genetic code. So that's what's in her genes, what's in someone's genes. So again, her genotype has both left-hand and right-handed genes, but her phenotype is only expressing the right-handed gene. So chromosomes, they store and transmit genetic information, and chromosomes are made up of DNA. So here we have a nice little image that kind of shows you this little X-shaped thing in the center is a chromosome, and it is, you can see how it's kind of taken apart to show the DNA, and then further taken apart to show the double helix of the DNA. And DNA are those long double-stranded or the double helix molecules that make up chromosomes. And we'll talk about these a little bit more later on, and you'll see some images of them later on in the video on epigenetics. Genes are a segment of DNA along a length of a chromosome. So when I say that she has a left-handed gene, that is a piece of a segment of DNA that's on one of her chromosomes. And again, those genes are made up of, it's a section of DNA. So here you have, the X shape of the chromosome. You have two genes on that section of DNA. So these two individuals, they differ in biological sex, they differ in race, they differ in a number of different things. Well, how genetically similar do you think that they are? So just kind of think about it, come up with a number in your head, what kind of percentage you think that they are genetically similar. So Justin Timberlake versus Beyonce. Well, human and chimpanzees are 98 to 99% identical DNA. So our closest primate relative, the chimpanzee, we share a vast majority of our DNA with. Individual humans, such as Beyonce versus Justin Timberlake, are usually about 99.1% identical. So that 0.9% is making up all the differences that you see in human expression, all the differences you see in the way humans look, all the way you see differences that you see in behavior that's related to genetics. All those differences are made up of a very, very small portion of the genetic code. So how we get the various genetics we get them from the parental sex cells. There's two sex cells, obviously, the sperm and the ovum. The ovum is for the woman, the sperm is the male, and each of those have 23 chromosomes. So each, the sperm cell and the ovum, each have 23 chromosomes, which is half of what a normal cell has. So when we say that you're made up of half and half of the sperm and the ovum, it's because, again, we have half is being provided, 23 by the sperm, and half is being provided by the ovum. When the sperm and the ovum join, this is what's called, when fertilization happens, this is what's called a zygote. And it's a single cell product of the joining of the sperm and the ovum. And this is an important term to remember, so I want you to really, write that down, think about it, because we're going to talk about that a lot once we get into prenatal development. There's a whole period that we talk about, the zygotic period of prenatal development. So that's an important term to flag. autosomes are the 23 matching pairs of chromosomes. So in this picture here, you see the pairs 1, 2, 3, all the way to 22. Those are matching pairs of chromosomes, the 22 matching pairs of chromosomes. That last pair you see down there are the sex chromosomes, X and Y. Obviously, this individual would be male because biologically male because they have an X and a Y chromosome. So those sex chromosomes, 2 X's are female, an X and a Y is male. So the ovum is always an X. The sperm is either contributing that sex chromosome of an X or a Y. So that differs between the two. Now, we can have multiple offspring. So when a sperm and an ovum join, even if it's just one ovum, we can have multiple offspring. If there are 2 ovums, we can have multiple offspring. And so that's what these images show here. So if we have 2 ovums, as you see at the bottom of this image, looking at D and E, we have 2 ovums there. Those are what are called dizygotic twins. Remember that term zygote I said, meaning the joining of a sperm and an egg? Dizygotic means there are 2 zygotes. Okay, so 2 ova equal 2 zygotes. Monozygotic twins, or what you'll see in the images A through C at the top, means there was 1 egg. There was 1 ova. And so we have 1 ova and 1 sperm that split into 2 separate organisms. But they are monozygotic twins. So that zygote splits into 2 cell clusters. Dizygotic twins are what we also refer to as fraternal twins. And they are just as genetically similar as any other sibling. So dizygotic twins are not any more genetically similar than they are to, say, their brother that's 3 years older. The genetic similarity are going to be the same because each one of those individuals was a separate egg and a separate sperm. Whereas monozygotic twins are what we refer to as identical twins. Those twins are actually genetic clones. They have the exact same genetic code at birth. And again, it is that one ova, that one fertilized ova, that one zygote that has split into multiple offspring, usually 2. But so again, we have the difference in the dizygotic, and it could be trizygotic if you're having triplets, that would be trizygotic. But again, it's about the number of ova that were present at the beginning. So remember, dizygotic twins are going to be no more genetically similar than they would be to any other sibling, whereas monozygotic twins are genetic clones of each other, naturally occurring genetic clones. So, how all these different things that make up all of our different behaviors and all of our different phenotype expressions are coming down to alleles. And each of 2 or more forms of a gene is located on the same place in the corresponding pair. So you have a pair of chromosomes, again, one from the paternal side, one from the maternal side, and those on the same place will have matching genes. So here we have in this image, we have P, A, and B, and these are just set up to kind of show you that these are matching alleles. We'll have P's in the same place, we have A's in the same place, and we have B's in the same place. In this image, capital letters indicate a dominant allele, and lowercase letters indicate a recessive allele. Okay, and so we'll break down the different genotypes that they're discussing there, the homozygotic, homo, hetero, and zygote. So when you have homozygous, it's alleles from both parents are alike. Okay, so for example, mom and dad both have brown eyes. They both contributed a brown-eyed allele, and so the kid has brown eyes. Another example, and that would be the case of the 2 capital P's here, okay? So both mom and dad had the dominant brown eye. Another example would be homo for the recessive allele, okay? So remember, homo means the same. So we have the same allele, and we have the little a's here. Both of those are recessive. Mom and dad both contributed blue eyes. So if you have blue eyes, that's a recessive allele, and you can only have blue eyes if both of your alleles are for blue. Okay, the same thing with left-handed. You can only be left-handed. That's a recessive allele, a recessive gene, if both parents are contributing that left-handed allele. Where heterozygous is where alleles from the parents are different. So when we see the big B and the little b here, that's what we're talking about. So the relationship between the alleles determines the expression. So if mom gave babies brown eyes and dad gave baby a blue eye allele, the baby will have brown eyes because the brown eye allele is dominant over the blue eye allele. So here's an example. Again, mom gave a brown eye, dad gave a blue eye, baby will have brown eyes. Okay, and this is what's called dominant recessive inheritance. So you have this dominant and recessive gene. You have both of them, but the dominant is going to be what's expressed in that phenotype, whereas your genotype has both the dominant and the recessive gene. Okay, so that again, that's dominant recessive inheritance. And what happens with dominant recessive inheritance is you can then have carriers, okay? So the genotype still has that recessive gene in it, and you can pass that gene on to subsequent generations. So, for example, in my own family, my dad has blue eyes, and I have green eyes. So that tells me that I have a green-eyed gene and a blue-eyed gene. Okay, I know that I have a blue-eyed gene because my dad has blue eyes, so he only has blue-eyed genes to pass on. So I know that he's contributed a blue-eyed gene to me. I also know that I have a green-eyed gene because I have green eyes, and that's what's being expressed. So I have dominant recessive inheritance, and I'm a carrier of that recessive gene, of that recessive blue-eyed gene, and I could pass that on to subsequent generations. My husband has blue eyes, so I also know that he has 2 recessive blue eyes alleles, and when we had a daughter, the only thing that he could pass on to her was a blue. So she had a 50-50 chance of either being green-eyed or blue-eyed, and it all determined by what kind of gene I gave her. Did I give her my blue or did I give her my green? So I was a carrier of this recessive gene. Britt has blue eyes, so I carried that recessive blue-eye gene. I did not express that blue-eye gene. I was a carrier of it, but I passed it on to my daughter, and so she has blue eyes because I was a carrier of that gene. So again, that's just an example of how that dominant recessive inheritance can work and how carriers can play a role in this. We can also have chromosomal abnormalities. Down syndrome is the most famous one, the people that most know about. And really what that is just an extra set of chromosomes. So here you can see here on the 21st pair, there are 3 chromosomes instead of 2. The other name for Down syndrome is trisomy 21, and that's because there are 3 chromosomes on the 21st pair, okay? So again, that's that extra set of chromosomes causes the genetic abnormality, which expresses itself with the symptoms and the physical changes of Down syndrome. The risk of Down syndrome goes up with maternal age. Maternal age is not the only contributing factor. There is genetics, and there are other things, but the risk definitely goes up with maternal age. At 20, mothers have a 1 in 1,900 chance of having a child with Down syndrome. At 35, it goes up to 1 in 300, and by 45, it's up to 1 in 30. So again, those chromosomal abnormalities can sometimes be related to maternal age. Not always, but in the case of Down syndrome, it is. So we've talked a lot about genes, a lot about kind of the nature part of our development, but let's kind of talk about how those genes can express themselves in terms of behavior and how we'll use these genes and behavior to ask questions about development. So one of the ways we do that is adoption studies. And what adoption studies can do is they can sort out what's attributable, what we can attribute to genetics versus what we attribute to the environment. It helps us parse apart what is nature and what is nurture, okay? So we can look at an adopted child and we can look at how they are similar to their adopted parents. That would be the environment. That would be the nurture. Versus we can look at how they're similar to their genetic parents. And that would be the nature. That would be the genes. And so again, adoption studies help us start to parse that apart. Genes can also influence the environment an individual encounters. So when we talked about Bronfenbrenner, remember we talked about children being both products and producers of their environment. So those genes can influence the environment that the individual encounters. That genes can kind of help create or produce those environments as well. We also look at genes and behavior with twin studies. So when we have those monozygotic twins, we have those identical genes. Remember, they are genetic clones, so they have the exact same genes. When we have those dizygotic twins, when we have those fraternal twins, they are no more similar than regular siblings. And so we can use this to study, say, personality traits. We use it to study many things, but one of the things we can use it for is to study personality traits or temperament. So again, we can kind of look at the contribution when we compare monozygotic versus dizygotic twins and see what role genetics is playing. Okay, we're going to, if monozygotic twins are more similar in something than dizygotic twins are, we're going to say that that is because of genes. Okay, where if dizygotic twins and monozygotic twins have the same level of similarity in something, we're going to say that's because of the environment or because of an interaction between the genes and the environment. When we're talking about these genes and the environment interacting with each other, one way that we talk about this is reaction range. Reaction range is the interaction between the genes and the environment, and it determines the upper and lower limits of development. So in this image here, we are looking at IQ scores and we're looking at environment and how environment and genetics are contributing to the expression of IQ, the expression of intelligence. So for each person, you see kind of a range above them. That is their individual reaction range. And basically that reaction range is set up by the genes. The genes say, okay, this is the range in which I can operate, okay? And what determines where on that gradient they are is the environment. So if you look at the blue group at the bottom, Jack, Jill, and Alice, they are all towards the bottom of their reaction range because they are in a deprived intellectual environment. Whereas Susan, Chris, and Lee in the orange stripe are kind of towards the middle or the average of their reaction range because they are in an average quality environment for intellectual potential. Whereas the individuals, Tom, Kimberly, and Jerome, are in the enriched environment. They're all towards the upper end of their reaction range. So again, the reaction range for each individual is inherited. That's in the genetics. What determines where in that range someone is the environment. So these things, again, are going back and forth with each other. Again, this is that bidirectional interaction that we're going to continue to talk about. And this is one way that we see it in terms of reaction range. We most typically talk about reaction range in terms of intelligence, but we definitely discuss it in other areas of development as well. Another thing we talk about when we talk about the nature and the nurture and biology versus environment is non-shared environments. So I have a brother who's 6 years younger than me. We have the same parents. We grew up in the same house. We went to the same schools. But that 6-year time difference gave us some differences in terms of our parents. It gave them some differences in terms of their finances. It gave them some differences in terms of what we were allowed to do with the rules. My favorite example that I love to give my mother and my brother a lot of trouble about is when I was little, I was not allowed to have pretty much any cereal that had sugar in it. So I was really in the Cheerios, Rice Krispies, if I was really lucky, Kix kind of cereal. Whereas by the time my brother had come around, my mother says that I had worn her down so much that my brother got Oreo O's, which is clearly a very high sugar cereal when it is named after a cookie. So those were ways that we had a non-shared environment. And this can be a source of developmental differences in children from the same families. Now, I do not think the fact that I had Cheerios versus my brother's Oreos really contributed into some major developmental differences for us. But the kinds of differences in rules that parents provide, the kinds of differences in finances, the kinds of differences in the way parents perceive their children can cause differences in children's outcomes. Individuals also experience environments differently. So you have all been in a situation where you perceive something happened one way and a friend perceived the exact same thing happened another way. You think someone was just being silly and your friend thinks someone was being mean, and you saw the exact same activity. Well, this can happen in families too, where you perceive the environment differently. And again, this can be a source of change and differences when we're experiencing environments differently or having different interpretations of those same environments. We also can get different reactions elicited because of that, okay? So because my brother and I had different genders, we often elicited different reactions from our parents. If I were to hit my brother, I didn't get in as much trouble with my dad, whereas my brother hit me, even though he was 6 years younger than me, he got in a lot more trouble with my dad. Whereas with my mom, we didn't have that. My mom, we were equally in trouble. If anything, I was in more trouble because I was older. And so we had different reactions. We had from different parents depending on different factors. So these are ways that which we had the same parents, we grew up in the same house, we grew up in the same community, we went to the same schools, but that we had non-shared environments. Everything was not the same for him and I. So again, remember, it's really important to remember that nature and nurture are really working together. This is not one or the other. They are working in tandem to produce these developmental outcomes. Remember when we talked about dynamic systems, that this web of development. And again, this is what's happening. Nature and nurture are working together. Development's occurring via this bidirectional, remember that's arrows going both directions, or co-actions with all levels of the environment and the organism. And so this is, again, going back and forth. And here's a really good image that I like, and this is from work from Gottlieb, and it shows individual development across the bottom. And you see these arrows kind of going to each level between environment, behavior, neural activity, genetic activity, and we see these interactions at every level of the organism. These interactions are co-actions with every level. So again, you're seeing this kind of web of development where nature and nurture are really working together to create these genetic expressions, to create the organism, to create behavior, that it really is this combination of both nature and nurture. So what I want you to do now is I'd really like you to actually pause this video and go and watch the video on epigenetics, okay? It's linked both on D2L, it's on the playlist on YouTube. The playlist on YouTube is actually two-part. If you follow this PBS link that's on D2L, it will be one continuous video. It's the same video, but please pause this video, go watch that now, and then we'll come back and we'll talk about epigenetics. Okay. So hopefully if you watch that epigenetic video, it really will make the next several slides make a lot more sense than if you watch the video afterwards. And again, highly, highly recommend you watch that video. It is the best thing I've seen on epigenetics ever. It's great. Okay, so epigenetics is the emergence of new structures or functions through this bidirectional interaction, again, between the genes and the environment, okay? So the environment's affecting the genes and the genes are affecting the environment, and this is going back and forth. Behaviors or structures emerge or fall out or self-organize because of this bidirectional interaction. And these emergences, again, are resulting from this co-action of the environment and the organism. So the genetics and the environment are going back and forth. These are not predetermined instructions. This is not necessarily in the genetic code. These are switches, as I talked about in the video, that are being turned on and off based on the environment. So epigenetics or epigenesis are these non-linear, non-obvious co-actions. So the example that they give you in the video is that of obesity and diabetes in mice, and how they have these genes being turned on and off of these mice. And they have the fur coat to kind of help determine as a marker so they can see who they've turned on these genes with and who they haven't. But again, this was being passed on through their mother's milk, okay? These mice were then predisposed to diabetes. They were less likely to develop in the mice that were predisposed to diabetes were less likely to develop it in adulthood if they were nursed by these lean, non-predisposed mothers. So again, they are genetically predisposed, but because these mothers had the switches turned off, so to speak, they were passing on those off switches to the mice that they were nursing, okay? Whereas mice with the lean genes were more likely to develop diabetes if nursed by these obese mothers. So again, the ones that weren't predisposed to diabetes, that weren't predisposed to obesity, were getting those switches turned on, essentially, by the obese mothers when they were getting the on switch information through the mother's milk. We also saw that for another example, is hypertension in rats was dependent on specific rearing or interactions conditions. So again, they were able to turn hypertension kind of on and off, dependent on how these rats were being cared for, how they were being reared by their mothers. Other examples is that timing is really important in epigenetics. The environment and the experience may have different impacts at different times. These on switches, per se, or these off switches, have to be introduced at specific times for this to cause this change of this genetic code. These are these sensitive or critical periods. Again, that are turning on or off the switches on top of that genetic code. Examples of this are studies with bobwhite quail. They were given different calls at different times and different visual stimulation at different times, and we saw different patterns of behavior dependent on that. We also see the same thing can be done in tissue transplants. If it's done early enough, that tissue can turn into a different kind of tissue, whereas if it happens later, it will never change. These epigenetic modifications, or these changes that we're seeing due to epigenetics, are these processes that alter the genetic activity without altering the DNA. Remember, epigenetics means above the genome, right? So these switches are turning genes on and off. The genes are still there. They haven't gone anywhere. It's just this gene is being turned on and off. Again, these are acting like switches, turning the genetic expression on or turning the genetic expression off. And this can be caused by a number of different environmental influences, such as toxins, experiences, behaviors, chemicals, nutrients. All of these things are things that can change this epigenetic code, that can turn these genes on or off. And again, these are more important in periods of high change. So we see that epigenetics is really, really important prenatally. We see that epigenetics is really, really important in infancy. Again, these are periods of rapid change in humans. We also see that epigenetics is playing a big role in the teenage era as well, in adolescence when we have puberty going on. And again, we have a large amount of change. We know that epigenetics is really involved in these disease processes. We know that it's also a process of typical development as well. So again, we see that these on and off switches are contributing greatly to the expression of genes, to the behavior changes, both in the animal models that are studied, but also you saw in those twins that were being studied. They had very different epigenetic codes the older they were than when they were young. And again, that's the influence in the environment on that genetic code. Epigenetic changes can impact development across generations. So there's an article that is linked in your on D2L that talks about how before a baby's even conceived, how a mother's diet can impact her offspring. There's also studies that famine of your grandmother can influence your genetic expression as well. We've also seen this with mouse color, fruit fly, eye structure, birth weight in humans, again, as being related to that famine study I was discussing. these epigenetic effects may be reversible. You saw in that video that they were using this to kind of turn off cancer in some kinds of ways. But we can also see that this epigenetic drift, where the adult twins were more dissimilar than the young twins. And again, that's the degree of chemical modification is a lot larger with age. And as the video discussed, that can be as much 4 times as much differently expressed genes between a pair of 50-year-old twins compared to a pair of 3-year-old twins. And remember, these are genetically identical twins, but those genes are being expressed differently because of this environmental influence. So that ends our discussion of the biological influence on development. Again, if you have not watched that epigenetics video, go watch it. Super, super important, super, super helpful. And really read that Science Direct article on epigenetics as well. It's also very helpful in understanding the processes here that we're talking about. Thanks, and I'll see you in class.